Hello everyone, how are we doing today? So today we're moving on into the next lecture in chapter 5 here, where we move into multi-locus extensions of inheritance. So this is kind of broken up into two videos because there's a lot to talk about. The first video we're looking at two gene interactions where epistasis plays a large part. So let's see what we have here. So here, two gene interactions and epistasis for inheritance here. Remember, this is still from chapter 5, which is just extensions of the typical Mendelian inheritance. Okay, so for this is when multiple genes lead to one phenotype. So multiple genes go to one phenotype. Example, my big fluffy hair right here that I need to get cut, but I haven't gotten it cut because we're currently in a pandemic. So one phenotype is my hair color. Now multiple genes could lead to that final output of that hair color. And there are also a lot of other factors that could be involved with that too. So you could do a little calculation on this if, it's not too crazy. So the number of genotypes possible is 3 to the n. Let's say n, where n is equal to the number of loci with 2 alleles each. So 2 alleles. So you can either one version or the other version. So let's say n is equal to 2. So 3 to the n is equal to 3 squared, which is equal to 9. So 9 possible genotypes if you have a loci with 2 alleles. So that's if you have 2 genes, 1 phenotype. But 9 different genotypes are possible within that. And I'll express that example a little bit more down here after getting a little bit into two gene interactions. I just wanted to show that equation there first. Okay, first here we have a simple reaction before we get into epistasis. Simple reaction, let's look at pepper color. So for pepper color, these example of peppers, you could be cream, orange, peach, or red colored bell peppers. So for cream, that's the recessive version. So recessive. So I'm using, so this is 2 genes, and there we have the C allele and the Y allele. Now the next color here is if C isn't the recessive form, this one could be either, and both Ys are still recessive. This one makes orange. Next one is if the C's are recessive and the Y is now the dominant form. And again, this can be any right there. Remember, underscore means any. This one leads to peach. And then the last version here is if you have a dominant form in each. So these 2 genes are interacting, and the way they interact changes the final color of that pepper. And then this is how you get a red bell pepper. So here we have 4 different phenotypes, but so we have 2 alleles here. So 2 alleles, and so n is equal to 2, so we should have 9 different genotypes. So here's 1, here's 1, 2, because that underscore represents 2. So here's 2, and then here's 1, 2, 3, 4. Each of those underscores would represent 2 different possibilities. So 4, 5, 6, 7, 8, 9. So there you can see where that equation comes from, where you can get that 9 if you have something like 2 alleles. So this is just a simple interaction. It's just how these products combine to then form that eventual color in that peach. And dosage, remember, if you have 1 form or 2 forms, sometimes can make a difference too. One thing I wanted to quickly mention here are these things called pseudo alleles. So remember, pseudo is false. So these are like false alleles. So these appear to be allelic, but actually are 2 separate but closely positioned loci. So we're going to be getting into linked genes in chapter 7, but you might have 2 genes that are located very, very close together. So they look like they might be allelic, but are actually 2 separate but closely positioned loci. So just wanted to bring those up here because sometimes they might get mixed up into these 2 gene interactions. All right, now the heart of this lecture is on epistasis. There are a bunch of different kinds of epistasis. We're going to talk about, I think, 3. We're going to talk about recessive epistasis, dominant epistasis, and duplicate recessive epistasis. So you can find tables that show the other forms of epistasis. We're only going to talk about these 3 because they're the main ones when you discuss them. Now, just as a reminder, let's go back to our typical cross. define epistasis here. So if we have a heterozygous cross, a dihybrid heterozygous cross, because we are talking about two genes now, what is the ratio we have here? We have that 9:3:3:1 ratio. If you do this cross, and it's typical Mendelian inheritance and epistasis isn't involved, you should get 9:3:3:1. However, if epistasis is at play, you don't get this ratio. So when I said you can look up the tables of epistasis, you can see the different variations of this ratio, and that can help you identify what the genes are doing in that individual. So what is epistasis? It's when one gene controls the expression of another. So this gene, depending on whether it's dominant or recessive, determines whether this gene is even presented, no matter what its dominant or recessive level is. So one gene controls the other. So you can have different levels of control when it comes to this. So the first one here is recessive epistasis, and this is one of the more common ones when you talk about them. So this is the epistatic gene. So here we have an epistatic gene and a hypostatic gene. So epi means upon, hypo means below. So the epistatic gene is upon the hypostatic gene. So here for recessive epistasis, one gene, which is the epistatic gene, controls the other gene, which is the hypostatic gene, and their interaction leads to one phenotype. So that's the general gist of epistasis. So recessive here, if the epistatic gene is recessive, the hypostatic is masked. So if this epistatic gene is recessive, you don't get expression of this gene over here, or it's masked. Not necessarily expression, it just might not present itself. So that's why one gene will then lead down this route for the phenotype rather than coming through here and coming down. So let's look at the example here. This example is using mice. I will attach a video to this in the description where I use Watson. Watson is currently sleeping on my toes right now, but Watson is a yellow lab. He's mixed with a border collie too, but he's mostly presents as a yellow lab. When he's a puppy, he had a black nose, but he has yellow fur. So how do yellow labs get a black nose and yellow fur? You can also have yellow labs with a brown nose and yellow fur. Remember, you have black, chocolate labs or brown labs, and then yellow labs. So this example here uses mice, but just imagine it's, brown here would be black labs, the lighter brown here would be brown labs, and then the white down here would be yellow labs. But just looking at, so coat color genetics is an example of this. So remember, epistasis here, one gene controls the other gene, and we'd be typically looking for that 9:3:3:1 ratio, but you don't see that here. So we did this cross, so this is that heterozygous cross right here. It might be hard to see, and I hate when examples use the letter C. So a heterozygous dihybrid cross, you should get a 9:3:3:1 ratio, but if you count all these up, it's actually a 9:3:4 ratio. So 9 black, 3 brown, 4 white or yellow, and that's not this. So something's going on, and 9:3:4 is recessive epistasis. So if you look at these results here, this one, everything's homozygous, dominant, and normal. If you look at this one, the first gene is recessive. So the pigment isn't made properly. So the second gene is made fine, but the first one isn't a dominant form, and that's what creates a little light brown mouse. Now, let's see what happens if the second gene is recessive. So down here, all of these second genes are recessive. You notice if all these are recessive, it doesn't matter what that first gene is. So because of that, that second gene is epistatic to the first gene. So the B is the hypostatic gene, and the C is the epistatic gene. So it doesn't matter what's there. It's going to be yellow fur color. Now in labs, you can kind of tell what this one is. So this one should be a black lab. This one should be black lab. This one should be a brown lab. So you can look at the lab's nose. So a yellow lab with a brown nose, you know, is recessive, and also has that for the epistatic gene. Yellow lab with a black nose, you know, is either one of those. So it could be homozygous or heterozygous there. So kind of interesting. And we'll talk about that later. So C allows for the pigment to be expressed in the fur. And that is why it is the epistatic gene. So another one I want to mention here is takes us back to blood typing. And it's very interesting. So it's called the Bombay phenotype. So the Bombay phenotype is when someone is phenotypically O type, but they actually carry the information for either A or B blood type, but they don't express it phenotypically. So the Bombay phenotype is a recessive epistasis. If this gene isn't expressed, the second gene fails to express. So here we call this compound H. So compound H, the dominant form, produces compound H. Think of it as an enzyme, whereas the recessive form doesn't produce it. That's the epistatic gene. The hypostatic gene is ABO blood typing. Remember, ABO blood typing, you can be A, B, or O, or AB. So if an individual is this genotype, they would actually present as O type. Isn't that wild? And if an individual is this, they would present as AB, because that, epistatic gene is presented. So this it gets interesting now, because this is how you can get on the Mari show. What cross do we want to do here to show this? Let's do a cross where we have an individual who has a Bombay phenotype, but they are AB. So this person, let's say they never got genetically tested, they didn't know they had this. This person's going to think they're O type, which is little I, little I. That's what they think. And then they cross with an individual who is homozygous dominant for that Bombay or compound H. They make it just fine, and this person presents himself as heterozygous B phenotype. Alrighty. So we have a B type that found an O type, so they think. So you do this cross here. Let me draw out the Punnett square. I'm not going to include these ones because they'll all be heterozygous, so we don't have to include them, and all of the offspring will be able to present their blood type just fine. So I'm going to write out the cross right now, and here are those results. Now look at this. If you were an individual who thought you were O phenotype and you knew your partner was B phenotype, and you had offspring that came out as, oops, this one should be a B, as A. So this one would be a what's going on moment. This one would be a what's going on moment. This one would be a what's going on moment, because how can you get an A type if you don't? So this, parent didn't have A type and you thought you were O type. So how you get A type into children? Whereas this one here, you wouldn't ask any questions. Be like, oh, B type makes sense if my partner's B type and I'm O. So this one makes sense. So a very interesting situation there. So it's, fun to think about. But it could happen. This is called the Bombay phenotype, where the, output doesn't match it. And how this works, remember when we talked about blood typing, you got the red blood cell, you got the carbohydrates coming off of it, and then you get that third one, and this is O type. What this compound H does is it allows for this one to be added. If this one isn't added, you can't get, so remember, so that would be O type, and then A or B type would depend on which one is added right here. You can't add either of these if you never add this one. So since this one isn't added, this is what then the Bombay phenotype shows on their red blood cells, and they act the same as an O type. Okay, so that was recessive epistasis. Like I said, you can talk the most about that one. Next one here is dominant epistasis. So if the epistatic gene is dominant, the hypostatic is masked. So the example I want to use here is squash color. So let's say we have a white squash, and there's a reaction, an enzymatic reaction that makes green squash. And then there could be another enzymatic reaction that then makes yellow squash. So how do we go from white to green? There's an enzyme involved. In this case, if it's dominant, that enzyme isn't produced. So it could inhibit an enzyme from working. If this one is recessive, little w, it allows that reaction to go, and it turns the squash green. And then there's an additional reaction here for enzyme 2, the big Y, little y, that would also block the reaction. So like I said, these can be inhibitors of reactions. Somehow they prevent this reaction from occurring. And then the recessive version allows that to go. It pretty much gives the green light, the recessive version giving the green light. So here, the epistatic gene is the dominant one. And the hypostatic can be masked in this process. So if you have a pepper that's, this one can just be an underscore. So if you have a pepper that's homozygous recessive and then dominant here, you'd have a green pepper. Whereas if you have a pepper that's homozygous recessive for each, that's the only way you can have a yellow pepper. And then to have a white pepper would be like this. So you can have 3 different phenotypes based on these different genotypes of how they react. So this is dominant epistasis, where the dominant one masks the hypostatic. So then duplicate recessive, this one, confusing terminology, but it's when one recessive can block the other recessive, which blocks the other recessive. Like I said, confusing terminology of how it phrases. So this is common in biochemical pathways where both enzymes need to produce one form of a phenotype. So there are 2 reactions, and it's the final outcome where you see a change. So either gene can mask the other. So here, gene A and gene B. So gene A can mask gene B, so it's the homozygous recessive A is epistatic to gene B. And then gene B, the homozygous recessive version, is epistatic to gene A. So that's what it means, duplicate recessive. So gene B could be epistatic to gene A, and gene A could be epistatic to gene B. So if we look at an example of snails here, so looking at the color of their shells. So you could have gene A makes, for white snails, or step A for white snails, and then B for white snails. So this is a step in the process, leads to then brown snails. So if it's recessive A or recessive B, neither, so if it's A, if this one's recessive, this reaction won't work, the snail will be white. If B is recessive, this reaction won't work, the snail is white. So means both of them are no color. So that's what I mean by duplicate recessive. So if this snail color here was this, that would be white because this one blocks. Same with this form. Doesn't matter that this one's fine, this one still blocks it. So duplicate recessive. So the only way you can get brown snails is this form right here. So the only way you can get brown is just each one works. So like I said, it makes sense that these are typically what you find in biochemical pathways. And later on, we'll talk about these when doing complementation tests to see if 2 genes are at 2 different areas. So say you get these 2 recessive white versions here. Are they in the same gene, or the same mutation, or different mutations in the same gene, or are they at 2 different loci? And by determining if they're at 2 different loci, you can then determine if the genes are different. It can help you figure out biochemical pathways and how things react with each other too. So yeah, that's all I have for this today. So we talked about duplicate recessive, dominant epistasis, the Bombay phenotype, and blood typing for lab color for recessive epistasis. I will link Watson's lab video, and then there will also be an example video going over recessive epistasis that comes up on the next published date. So remember, these videos are published Mondays and Thursdays at 9 a.m. for this genetics course. But that Watson Lab video is already made, so I'll just link it, and it'll be part of the playlist if you look there too. All right, and then we talked about some simple 2-gene interactions too. All right, so next video, we're going to continue multi-locus extensions. So next lecture video, continue in multi-locus extensions and getting into polygenic inheritance. All right, where many genes affect the outcome. Hope you all have a great day, and then see you all next time.